Marta comes to us from Rome, which is hard for me to believe anyone would willingly leave. Through her tenure at the NINDS, working with Jeff Dunn, she got an early introduction to 7 Tesla, pushing the spatial resolution limits and looking to mitigate physiological noise. She came to us in 2011 and has since been working on the brainstem and deep nuclei connectomics. And this is a place where she has plenty of need for both spatial resolution and mitigating physiological noise.
Thank you so much. And yeah, it's great to be here and to listen to these stories about the 7 Tesla. My story, I think I basically had a crush for the 7 Tesla on day 1 of experiment 1. I moved to the NIH. I was a postdoc in Jeff Dunn's lab. He's here and he'll give a talk soon so he can prove or disprove what I'm saying. And originally I was supposed to run some EEG fMRI experiments at 3 Tesla, but Jeff made also available the 7 Tesla, and I was very curious about it. So I ran a quick pilot study on 2 subjects investigating the effect of behavior, like having your eyes closed or open, either with or without fixation, on the underlying resting state signal fluctuations. And I still remember that after transferring the data and just running a quick, putting a seed in 1 voxel in the visual cortex, I could observe some results at the single subject level. That was the beauty of the 7 Tesla. And the results were consistent across both subjects. And basically I observed that with eyes closed, the level of resting state fluctuation is higher than with eyes open, either with or without stimulation. And that the visual stimulus, the evoked activity in green overlapped in some regions in yellow with underlying resting state fluctuations. And of course, we performed a study on a group of subjects. Here I'm showing you the Fourier transform of the signal time courses in the visual cortex. And the fact that we had a modulation by behavior of the resting state fluctuations indicated that at least part of these fluctuations were related to neuronal activity. And the linear superposition of evoked and spontaneous activity had implications for task-related studies. So basically, in order to characterize evoked responses, it was important to properly characterize also the underlying spontaneous fluctuations. And this was my first experiment at 7 Tesla. We ran many more other experiments. Basically, I only worked at 7 Tesla when I was in the Jeff lab, and I'm still working at the Martinos Center mainly on 7 Tesla. So of course, at 7 Tesla, you have high sensitivity for signal of interest, but also as mentioned by Christina, you have higher sensitivity for physiological noise. And there was this debate, are these resting state fluctuations just an artifact of physiological origin, or they contain some neuronal information? And so we modeled, we developed new ways of modeling some physiological noise sources, and we measured the variance explained by different noise sources in both fMRI data at 7 Tesla in the visual cortex. And yes, indeed, the physiological noise and also thermal noise explained a big chunk of this pie chart, but we had 21% of the residue that most likely was associated to neuronal activity. And we also looked at the phase data. This was magnitude data. And so if this is the pie for magnitude data that I just showed you, in the phase data, we had the residue was much smaller. We had only 5% of residual fluctuations, and mainly physiological noise accounted for most of the variants, mainly low frequency drifts and respiration. And so to dig out this 5% out of this noisy data, we had to work hard and remove this physiological noise contribution. So this was like the original phase time course. And after removing phase wraps, we obtained a better time course, still with a noisy time course. And then after removing a polynomial, after spatial polynomial fitting, which mainly accounted for respiration, we obtained a time course in the phase that closely resembled the magnitude time course, and also the stimulus regressor. The phase activity map, both during task and at rest, co-localized with the magnitude activity maps. But more interestingly, the phase activity maps were not confined to larger vessels as originally thought, but were spread all over the cortex. And this indicated that the presence of oriented intracortical and pial veins that could underline these effects. And we took a step further to quantify susceptibility changes and oxygenation from this data. So from phase maps, we quantified frequency shifts, and this is the map. From frequency shifts, the susceptibility changes. And then from larger veins like sagittal sinus, we could extract from susceptibility changes the fractional oxygen saturation changes. And interestingly, those correlated with the magnitude changes in the cortex. So I think this was an exciting experiment. And yeah, I wonder, whenever I was preparing these slides, I wonder why I was not continue working on this project. Kind of moved to the Martinos Center, to the MGH. So it was 2011. I was 7 months pregnant with my second baby, and still Bruce and Larry hired me as a junior faculty. Yeah, most of you know that this is not the view from my office. You need to go to the Museum of Science. And at the Martinos Center, I took 2 research pathways. One was to continue working on physiological noise, this time as an opportunity to investigate cerebrovascular brain parenchymal compliance. In the extramural world, you really need to be pragmatic if you want to get the grant. And then also explore new topics like brainstem neuroanatomy, histology, and brainstem-related diseases like disorders of consciousness and Parkinson's disease. So with the first topic, the idea was to look at the MRI pulse waveform during cardiac and respiratory cycles. And from this, extract some indicators of compliance that would directly measure changes in blood volume rather than blood velocity, as for example in Doppler techniques. And so I proposed the pulsatility volume index related to the signal, to the ratio of the signal during systole divided by the signal during diastole. But I had to constrain the sequence parameters to produce a contrast dependent primarily on the spin volume rather than spin velocity effects. And from a simulation, there is a critical speed, that's the speed beyond which the signal does not depend on spin velocity anymore. So basically, we had to choose the critical speed below the blood flow velocity of interest, like in the carotid arteries or in larger arteries. And also worked a little bit on enhancing the endogenous contrast. So in this regime of fast-flowing spin, the Ernst angle is not the best angle. Instead, the 90 degrees gives a much higher signal. As we also demonstrated by this experiment, you can see that the carotid arteries are enhanced using a 90-degree flip angle compared to lower flip angles. Oops, sorry. Yeah, and with this, going back. I had some movies here, yes. And so with these sequence parameters, we were able to produce some movies of the pulsatility effects in the carotid arteries, in the scalp arteries. Also in the anterior cerebral artery, in the middle cerebral artery. Interestingly, also in the larger veins, like the sagittal sinus, you can see here the pulsatility, and probably this is an indirect effect through the CSF. And here in the CSF, so you can see how the CSF is pushed out from the lateral ventricles to the third ventricle, and this agrees with the Monroe-Kelly doctrine, for which we need to have a constant intracranial volume. So for every increase, systolic increase in arterial blood volume, the blood in veins and the CSF are pushed out from the cranium. And from these movies, we extracted the pulsatility volume index at the voxel level in the carotid arteries, cross-sectionally and in different segments of the carotid arteries, and we found a difference in compliance between different branches of the carotid arteries. And also we provided a preliminary validation of the pulsatility volume index as a dynamic compliance indicator during breath holding experiments. So you can see that during breath holding, this indicator went up in both our subjects. And then I reached my second career pathway and the last topic of this talk. So I noticed that brainstem imaging was really an underserved area of research compared to the cortex. So if you look at the cortical atlases and the connectomes that are derived from these atlases, you can see that the cortex is subdivided in more than 100 regions. Instead, the brainstem is assumed to be a big lump of all the brainstem nuclei. And the brainstem here is just represented by this green rectangle. Instead, the human brainstem is it's a small region. It's slightly bigger than a thumb, but it contains more than 170 nuclei. And if we count the subregions, there are more than 300 subnuclei. And each of these are involved in several vital functions, such as sleep, wakefulness, autonomic regulation, sensory motor, pain, and emotion. And the in vivo brainstem nuclei localization is mainly limited by the diminished gray-white matter contrast in the brainstem compared to the cortex. If we zoom in at this level, you can appreciate how homogeneous the contrast is. And the in vivo nuclei localization is guided by ex vivo, by mainly visual extrapolation from ex vivo atlases with limited precision in research and clinical studies. For example, here, there is a deep brain stimulation electrode for Parkinson's disease, and it's not known exactly if it targeted the pedunculotegmental or the neighboring cuneiform nucleus. Or in coma, we observe some microbleeds, but it's difficult to predict if the subjects will wake up if we don't know exactly which nuclei are affected and also how much. So in 2012, I started looking into different contrasts at 7 Tesla with the purpose of identifying brainstem nuclei. I got slightly better result with T2 star imaging than with the T1 weighted images. But the most encouraging result came when, with a visiting collaborator at that time, Nicola Toschi, after several weeks and months of optimization, we were trying to make diffusion work at 7 Tesla, and not many people were doing it at that time. And even now, I feel not many people use diffusion at 7 Tesla. But we were very interested in the high resolution and the ability of having multi-contrast images with the same bandwidth and with the same spatial resolution. And then after these months of work, we got reasonable images, and I think I had a vision. So it was not a craving because I was not pregnant at that time, but I saw in the lower part of the brainstem, I saw these gray matter areas, which are the brainstem olives, also called the olivary nuclei or olivary bodies, which are involved in multifunction. And this was the first nucleus I noticed. I remember Larry told me, oh, you should do 1 millimeter resolution. This was 1.7 millimeter resolution. I could not do 1, but I could do 1.1 millimeter resolution because of the acoustic resonance effect. I couldn't push the echo spacing further. But still, in these images, I was able to identify several brainstem nuclei, and I also considered a T2 weighted image for its high contrast for additional brainstem nuclei. So I started segmenting, semi-automatically segmenting both FA maps and T2 weighted echoplanar images on a single subject basis. Again, the beauty of the 70 Tesla was getting results on a single subject basis. And after a fine and diffeomorphic registration to stochastic space and computing the overlap of these regions across subjects, I was able to compute a probabilistic atlas label. At first by myself, and then with the help of Guadalupe and Kavita, who are also here today, and Nicola and Christian, we were able to generate an atlas of 21 brainstem nuclei involved in arousal, autonomic, and sensory motor function, and we are currently delineating 10 more nuclei. We are evaluating the functional connectome of this brainstem nuclei with 1.1 millimeter resolution fMRI at 7 Tesla, and also the structural connectome using probabilistic topography based on 1.7 millimeter resolution diffusion imaging at 7 Tesla. We are validating our macro scale in vivo atlas with histology, and we're bridging a gap between the macro scale and the sub-micrometer histology images by using a mesoscale ex vivo MRI, and we are validating both the nuclei location. So for example, the medial and the lateral parabrachial nuclei are adjacent to the superior cerebellar peduncle, as in ex vivo and in the histology. And we are also trying to relate some diffusion properties with, for example, fiber orientation maps computed from the myelin stain in Yes, mine still. We are validating our connectome by comparing it to literature reports in animals. We extracted this connectome from the rodent brain architecture management system database, and we are working on this comparison, and we'd like to add data also from primates. With the help of our clinical collaborators from the MGH, Brian Adlow and Alexander Vidanovic, we're applying the atlas to traumatic coma patients in the acute phase. The patients we considered all recovered consciousness within 6 months, and our brainstem nuclei analysis of microbleeds indicated that there was no single arousal nucleus that was lesioned in all the cohort, but a combination of nuclei. Indicating the presence of redundancy mechanism, arousal mechanism in humans. We are also applying the atlas to premotor Parkinson disease. We have a cohort of REM sleep behavior disorder patients. These patients have up to a 90% risk of developing a neurodegenerative synucleinopathy within 14 years from their diagnosis. So are a good window in the premotor Parkinson disease. And our structural connectivity analysis shows changes in the connectivity of specific brainstem nuclei that are in line with animal and with ex vivo human studies. All this is part of the Human Brainstem Navigator project. We are really working hard to release the Brainstem Nuclei Atlas, and we hope it will be a useful tool in conventional research and clinical studies of brainstem function and structure in both health and in disease. Yeah, so this is my plan for the future. I recently got this comment from a reviewer on a grant application. And he says the PI lacks prominent and unique neuroimaging expertise in high field and high resolution imaging. So my plan is to work on this. I really thank you so much for getting and making the 7 Tesla to work. I thank my advisors, my team, collaborators, and thank you for your attention.